techniques, applications, and prospect of human molecular genetics and cytogenetics. Today we have distinguished speaker, Professor Dr. Ashoke Pal. First of all, I would like to request Revered Swami Vedanudagandaji Maharaj to felicitate Professor Ashoke Pal. Now, I would like to request Maharaj to deliver the welcome address.
A very good afternoon to all of you. Today, first of all, I offer my respects to the speaker of today's seminar. I offer my respects to all the teachers present over here, staff members, and dear students. Today, we have been very happy to organize a one-day lecture on techniques, applications, and prospects of human molecular genetics and cytogenetics. It will be delivered by Professor Dr. Ashoke Pal, Professor Human Genetics, Center for Genetics and Genomics, Mahatma Gandhi Institute of Medical Sciences, Sevagram, Maharashtra. We are very honored and we are very glad to have Sir amidst us, who has taken pain to come to our college amidst his very busy schedule, and we are sure that this session will be a very enriching session, and we are always trying to organize seminars and workshops so that our students can come in contact with scientists and academicians and have exchange of ideas and academic enrichment. We are very grateful to all our teachers who are helping us in organizing this seminars and workshop. Today, for this seminar, we have Dr. Ajay Mullik sir, who as the coordinator for this seminar, and Dr. Bulganin Mitra sir is also there. So he has also been functional in inviting sir for this seminar. So we are also grateful to sir. And so without wasting much time, I, on behalf of the college, the Ramakrishna Mission Vivekananda Centenary College and the Department of Zoology, I extend a warm welcome to Professor Ashoke Pal sir for coming to our college and delivering this wonderful seminar lecture. And we would request our students to be attentive, and you should interact with sir after the talk. So you note down the major highlights of the seminar, and you are free to ask questions to sir after the seminar. That is how this kind of sessions will be interactive and useful to all of you. So with these few words, I again extend a warm welcome to our respected speaker for today's seminar, and to all our respected teachers and dear students for coming to this seminar. Thank you all. Namaskar.
A brief introduction about Sir Professor Dr. Ashoke Pal. Actually, Professor Ashoke Pal is a distinguished geneticist and academician with over 3 decades of experience in human genetics, cytogenetics, and molecular mutagenesis. He earned his BSc Zoology honors and MSc Zoology from Calcutta University and completed his PhD from Bardwan University. His research career began with a junior research fellowship at Indian Council for Medical Research (ICMR), and also he performed various role in as a research assistant, senior assistant research officer, CSIR scientist, and finally he joined Mahatma Gandhi Institute of Medical Sciences, Sevagram, since 1992, and retired as a professor, and till date actually he is also associated with that particular institute, and he has published more than 64 publications in national and international reputed journals. So, and he also uh, conducted various workshops and symposiums, and also undertake various research projects funded by different ministries. So this is actually very brief biodata. Actually, now I would like to request Sir to deliver today's talk.
Hello, 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 hello.
Hello, hello.
Hello, good afternoon everyone, and Swamiji also. Today I will talk on human genetics. Of course, you can take as a just motivational. Why I am telling motivational? As I was introduced with Dr. Ajay, that I was working in a medical college as a professor, and I started my career from Calcutta University, Bardwan University, and animal genetics, Drosophila genetics. But pardon? Hello. Sound. Hello, 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 hello. Hello. ওইখানে নো প্রবলেম ঠিক
সমস্যা
হ্যাঁ হ্যালো I shall specifically talk on human genetics and medical genetics. And I was telling that is a motivational, it is motivational because of the fact in our Indian colleges and universities, animal genetics, Drosophila genetics are being taught, but human genetics is not. Nowadays, in Medical Council of India, they have started compulsorily human genetics and molecular genetics division, department. But the fallacy is this, they are not getting trained human geneticist or medical geneticist. So here I will talk on human genetics and medical genetics. Of course, I will go very basic level only to create interest or motivate you people. This is my actually working place. It is near Nagpur. Spot is Shavagram. It is basically a tourist spot. Gandhiji made his second ashram after Ahmedabad, that is the Shavagram ashram. There the institute, very historical importance. You see, the title I have put here is Role, Relevance, Scope of Medical Genetics with special reference to genetic counseling. This is because of the fact I usually give lectures in medical colleges. So relevant to that, I put this title. But as your today's topic is applications, techniques of human molecular genetics, it is of the same. Only thing is its alternative title. I will cover techniques, applications, relevance, essence, whatever of medical genetics or human genetics, but it is an alternative title. If you see the human biology, that is anatomy, physiology, biochemistry, structural aspects, all are the game plan or logistics. I use the term logistics of our human body. Everything is planned and managed by the chromosome, gene, genome and DNA. Total human body activities, anatomy, physiology, biochemistry is controlled by chromosome, gene, genome. If I go at the base level, if ask a school student what is genetics, simple tell, science of heredity. But it is exactly not covering the whole story. This is because of the fact everything is not hereditary. Say somatic cell genetics, non-hereditary cancers, epigenetics, all are not hereditary. So if we define genetics as a hereditary, it is not completely correct, is incomplete. Rather, if we tell what is genetics, rather you complete it is a science of DNA. If you tell it science of DNA, it cover all aspects of genetics and science of heredity also. Simple thing what I pinpoint here is genetic science of heredity. It is all right, but it is incomplete because it is not covering the somatic cell genetics, or cancer is a somatic cell disorder. It is not covering epigenetics. It is not covered. So if we put science of heredity second position, but first position is science of DNA, that is the genetics. Science of DNA is genetics, followed by science of heredity. In our body, 100 trillion cells are working in a coordination. And how the coordination is being made, it is difficult to understand. Not known. That is what basically chromosome dynamics and the logistics. Still science is trying to search out. That is why if you go through the papers, research papers, you will see what they are telling. At the conclusion, more works to be done. Most of the papers, more works to be done. Because of the fact, still till date, we don't know the chromosome biology totally, or logistic. What we know, logistic, that is how do the cells manage all these physiological activities, structural activities. I will come. Not known. In fact, 5% of the physical system is known to us. Only 5%. And less than 5% is of the biological system is known to us. We don't know. Okay? And you just read that trillions of cells work in coordination and orchestrated way, making a symphony of systems, all under the direction of supervision of chromosome, gene, genome. Everything is directed, composed, regulated, chromosome, gene, genome. Okay? So this will give you the impression what is genetics. It is not that you people are reading genetics, I am genetics. It is. It is. Of course, one thing I am telling you, as I was working in a medical college and I was involved with the diagnosis of disease disorders. So my lectures will be targeted with human chromosome, gene, genome. Okay? And you see, if you go through 3-4 lines here, if you go to 3 or 4 lines here, that is all human disease is genetic in origin, basic etiology, ultimate diagnosis, prognosis, final treatment of diagnosis, understanding of genetics is a must. That will give you the impression of utility, essence of human genetics or medical genetics, to understand the human biology as well as disease diagnosis and prognosis and treatment. This is cell, you know, nothing to tell about. But the thing is, one thing I will mention here, whenever we read in the textbook, these are cell organelles. Go through to understand the logistics of the cell organelles. Logistics, how do they manage? No, science doesn't know now. Only partially knows. This is the metaphase. Okay, I use the term dynamics and logistics. If you know the chromosome structure, I know you know, but it is very systematically I put DNA, this histone octamer wrapped by DNA, and there are different types of histone protein making 30 nanometer scaffold and making chromatin, and ultimately 600 or 700 nanometer chromosome. Okay, that in the textbook you read it. Okay, no problem. And if some question is there, you just vomit it very nicely and score marks also. But that is not the end of the story. The question is, why do the DNA wrapping the histone octamer, and histone octamer they wrap 1.7 times, and there is only 146 pair base pair. Simple thing I am telling you, if I am going to tell on this structure, it will take hour or more. Simple, only you note down here that DNA wrapped the histone octamer and making solenoid structure, chromatin, and ultimately even getting the chromosome. The chromosomal logistics and natural logistics, here you can say, I am telling you, human has 46 chromosomes, correct? Fine. And genome base pair, 3.2 billion base pair. Okay. Length of the DNA in a diploid cell, 2 meter. Do you know the length of the DNA you have? What is the length you do have? Okay, I am telling you a very simple way. If you stress, there are trillions of cells, 100 trillion cells in your body, and if you just ligate and stress the DNA, you can find the length of DNA. It will reach to the sun and back to the earth 100 times. Okay? It will reach to the sun and back to the earth 100 times. And very okay, just here to mention, I will details. Here is male karyotype and female karyotype, 46X and XY. That you know it. I am not going to details. Okay. And cell cycle and cell divisions. Actually, cell cycle and cell division is a major part and very critical part in human genetics, because if you have some scratch or you have a cut here, within for 24 or 32 hours it is repaired by cell division, mitosis. Okay, and basically genetics is meant for reproduction. Genetics mean for reproduction, and reproduction mean cell division, that is mitosis and meiosis. If something wrong goes in the mitosis meiosis process, there may be cancer. If something problem in the mitosis meiosis process, some syndrome patient will be produced. I will come all this syndrome patient and recurrent abortion. So many bad obstetric histories. Simple thing, why I am just putting these things as cell division? That is, cell division is playing a vital role in genetics. I give example, if it is cut, repairing by mitosis, genetics is reproduction, and for basic unit of reproduction is gametes, sperm or ovum, and sperm ovum is produced by mitosis and meiosis. And something wrong in the mitosis meiosis process, that will give rise to some disease or disorder. Well, we generally talk genetics, human genetics, medical genetics. In fact, what I put here is gem in science: genetics, epigenetics, and mitochondrial genetics. What genetics, whenever we use the term genetics, one thing click in our mind: simple Mendelian genetics. One pair of chromosome, homologous chromosome, and what is called biallelic expression. One pair of chromosome, homologous, biallelic expression. This is conventional way. Genetics means this one. But no. Another very critical chapter, of course, last 15-20 years it is coming up very widely. It is epigenetics, where monoallelic expression works. You need not biallelic expression, you need monoallelic expression. In case biallelic expression is there, that will be a disorder. That is the nature's logistics. Why this is made, I will come. And next chapter is mitochondrial genetics. Mitochondrial genetics playing another very important role. Generally, we don't much bother for this mitochondrial genetics, but without mitochondrial genetics, our conventional genetics will not work. It is simply complementary and supplementary. Mitochondrial genetics, it is supplementary or complementary to our main course of genetics, or Mendelian genetics. So, gem in science, I mean genetics, epigenetics, and mitochondrial genetics. One interesting, you go to doctor, when they cannot diagnosis, they cannot make any diagnosis, what do they tell? Oh, possibly it is genetic disease. Possibly it is genetic disease. But nowadays genetic disease we can pinpoint with sequencing, my mutation study, because we have NGS system, we have NGS system. We sequence it, we pinpoint the single base mutation. So what doctors nowadays, अरे डॉक्टर साहब हम तो सीक्वेंसिंग करके आया, कुछ म्यूटेशन तो नहीं है. What do they do now? Oh, possibly it is epigenetics. Possibly it is epigenetics. In epigenetics, the interesting part is there is no mutation, there is no change in the DNA. Okay? There is no change in the DNA, only change in the expression level. You got my point? No change in the DNA, expression the DNA level, expression. How? How? It is blocked. Nature's in his own way, in his own logistics, blocked that particular gene or genome. So it is not expressed. So what is happening? It blocked one unit and another one unit is working. So it is monolithic expression. Simple thing what I told last 2-3 minutes. It is genetics, epigenetics and mitochondrial genetics. All are complementary or supplementary. The conventional is biallelic expression and epigenetic is monolithic expression, not change in the DNA, only expression level, it is blocked. I am not going to this thing. Yes, I have already talked relevance of mitosis and meiosis. Here what I have shown that for repairing mitosis is there, very simple thing, but for gametogenesis, mitosis followed by meiosis. Mitosis followed by meiosis. But one interesting thing I would like to pinpoint here. In male and female, both we term as gametogenesis. In male, it is spermatogenesis, making the sperm, with the division of stem cells, mitotic division, followed by meiosis division, and making sperm. How many? From 1 to 4. 4 haploid sperm cells. Why I am telling, in the next slide I will clarify it. Here only to cell mitosis, stem cell mitosis followed by meiosis. In case of spermatogenesis, they making 4 sperm. And spermatogenesis takes place at the adolescent period or puberty and lifelong. But here it is something different. I put both mitosis and meiosis in male. Both are human beings. Started at puberty. Lifelong process. 1 to 4. But in case of oogenesis female, same thing is happening, mitosis and meiosis. But nature's logistic, I was the term nature's logistic. Nature's logistics mean nature's management system, which we don't know. What is happening? When the fetus in her mother womb, oogenesis starts. Try to understand the contrast. I told in male it's about puberty, but here in case of oogenesis in female, when the fetus in her mother womb started. And first meiotic prophase is completed when she is in the mother womb. And arrested at first meiotic phase, diplotin stage, and so on. And first meiosis is completed just before the ovulation. And subsequently, Second meiosis is completed after fertilization when it meets the sperm. I am telling you this just step by step, but I am making the contrast with the gametogenesis, spermatogenesis and nature's logistics. I am just repeating again, in case of male, germ cells, mitosis, followed by meiosis, at puberty making 1 to 4. But in case of female, it starts when the baby in her mother womb, first meiosis is completed, and ultimately during ovulation, first meiosis is completed, and second meiosis completed after fertilization when meet the sperm. An interesting thing, what I am pinpointing here, there are how many sperm? 1 to 4. But here it is not. 3 polar bodies released, only 1. How does the nature select good and bad and this thing? Still it is a riddle. Simple. Take the message. In case of male and female, common name is gametogenesis, divided spermatogenesis and oogenesis. Both follow the mitosis and meiosis, but time scale is different. And the unit, 4 unit and only 1 unit, it is different. You can see the polar body in female, it is not. And more interestingly, that start at mother womb. Okay? Not only that interesting, other thing I will come in the subsequent slides, but rather as I started it, rather I should complete it now. Male is XX, male is XY karyotype, female is XX, 2X. What is the functions of chromosome? Making protein. Chromosome means DNA and something, making protein. What will happen if 2X, the female candidate making the protein, and you people, male, and protein from 1X, the amount is half, no? Got it? Got it? You have X chromosome, 1. Female is 2X chromosome. If they are making protein, we know chromosome or DNA from making protein, they will the double amount of protein. आप लोग का हाल क्या होगा? हाँ? ठीक है, कुछ नहीं, आप लोग ठीक ही है. Why I am telling this thing? One of the 2X chromosome in case of female is inactivated. What is called? Bar body. Okay? What is called bar body? This is bar body. The interesting part, and again I am telling natural logistics. If it is bar body during oogenesis. Bar body means that is chromosome DNA is totally packed. You cannot open it up. Correct? So during oogenesis, some problem will be there? No. Nature's made its all problem solution in its own way. Bar body, during meiosis process, it is reactivated again. It was inactivated, making a bar body, but during meiosis process, in case of female oogenesis, that inactive X reactivated, taking its own form. Okay? Then segregation of chromosome happening, that is no problem. If bar body is there, it cannot make a proper segregation. It is reactivated, making its shape and segregated in a proper way. ये nature's logistics, you see. अगर ये bar body रह जाता है, वो स्पिंडल फाइबर्स को कैसे खींचता उसको, सेपरेशन नहीं होता. Okay, natural logistics. Not only this, I will come to the next chromosome slides. Barr body, what is the location of barr body? Location of barr body? It is located or attached to the inner wall of the nucleus. Inner wall of the nucleus. I will show in the slides. The thing is, why it is attached to the wall of the nucleus? तुम भाई अभी काम का नहीं है, प्रॉब्लम मत क्रिएट करो, साइड में चले जाओ. Simple thing. Nature's made it in this way. Not only barr body, all the chromosomes in our body, not randomly distributed. It is not sprinkled. It is not randomly distributed. In a cell, all the chromosomes are placed in a specific location, specific manner. Not only this, in our body, I told you how many trillions of cells we have, and our cell types, 220 types of cells we have in our body, and 220 types of cells making different types of tissues or organs. They are performing different kinds of functions. Depending on the functions of the organ or tissues to be performed, chromosomes location in this particular cell or tissue. But simple fact is, depending upon the functions to be performed by the tissues, chromosome takes their place, or cells take their placement in the cell. Of course, if that placement is not properly placed in the cell, some disease and disorder will happen. Again, message is this: when we make some chromosome metaphase cell, we see chromosomes are randomly distributed, but it is a preparation problem, but it is not. Chromosomes are very specifically in the cells and specific site, specific manner, and different tissues, different cells in specific types, depending on the functions to be performed. And if something wrong in the placement of the chromosome in the cell, face the music, disease and disorders. Well, this is another very interesting thing. This is I am showing because of the fact in my subsequent slides, syndromes, this will come very regular way. Male is XY, no? And Y chromosome, that is SRY gene. It is X chromosome, that is SRY gene in Y. And this is spermatogenesis controlling gene. You are male because you have testes and because of this SRY. And spermatogenesis because of spermatogenetic gene. This is XY. What is happening during gametogenesis? Crossing over recombination. It is a natural process. It is a process of evolution. For evolution purposes, we need this, or variation purposes of chromosome, we need this crossing over recombination. Sometimes, due to some problem in the recombination, of course, it is all man-made. Man-made in the sense, what I meant to say, that is, our lifestyle. We are exposed to so many chemicals and so many drugs. Medicine is helpful, but it has some limitations. So what is happening, that SRY gene crossed over to X chromosome. This is X and Y, SRY in Y chromosome crossing over, and ultimately you are getting Y chromosome lost the SRY and X chromosome gained the SRY. So there is a problem in the sperm. X having SRY, very unusual, due to problem in the crossover. Why I am doing this thing in syndrome patient, I will explain this thing. Just look, I am going to discuss. The message, everything our body, crafted, programmed, everything is edited by chromosome gene genome. Just for your information, which is that lady? Huh? Rosalind Franklin. She got Nobel Prize? मिला नहीं, मिलना चाहिए था. Huh? Basically, she was the lady working with X-ray and X-ray diffraction. Anyhow, bad luck to lady Rosalind, she did not get. Rather, there is some stories also, that is not our discussion point. How many types of chromosomes? Metaphase? Sorry. Metacentric? I am telling, पूरा में थोड़ा मेरा metacentric, submetacentric, acrocentric, telocentric. Correct? I intentionally try to learn from you or listen from you. In human, no telocentric chromosome. In human, only metacentric, submetacentric, and acrocentric chromosome. No telocentric chromosome. Telocentric chromosome you can find in case of mice. Very common mistake. Okay? And I am not going to tell metacentric, submetacentric, and acrocentric. You know it better than me. You just see this, that how nature had made this genome. And as I told you, that 100 trillion cells, 220 types of cells in human body, 46 chromosomes, and 52 million nucleosomes. And this is base pair, and in one cell, 2 meters of DNA. And ultimately what I told you, that is total length of DNA in a human body reached to the sun and back to the earth 100 times. Okay? Okay. It is written in the text. Again I am referring to the logistics. When there is a cut, that is to be repaired by mitosis. The process, the DNA replication and other processes take place. If I take example of WBC or lymphocytes, within 24 hours they just unwrapped, unwinding the DNA and replicate and making complete the all process. Just think of how this length of DNA they completed within a stipulated time of 24 hours. That I told you. At the same time, that your length of your DNA, nuclear DNA, I mean to say, nuclear DNA, this to sound 100 times, but we never tell about the mitochondria. Mitochondrial DNA, it also reached to sound back to Earth 100 times, 100 not 1 times. Interesting part is this: in your cell, chromosomes are paired and paired, one homologous pair. But what about mitochondrial DNA? You have some people know microbiology? No. Mitochondrial DNA is different from the nuclear DNA. Mitochondrial DNA is a extra chromosomal DNA, extra nuclear DNA, and it is a circular DNA. Okay? It is a circular DNA. And if you take the history, that is originated from the bacteria or something. Okay? Okay? But one interesting thing is there, that is to be noted. In case of eukaryotes, if we take human genome, it is 3% of the genome is functional, we consider. That is eukaryotic, euchromatin. And 97% is not working. That is the concept. That was the concept, rather I should say. 3% is functional, that is gene, making protein polypeptide. And other is non-functional. Of course, that is not correct. In the recent years, we can define that no portion of genome is not useless, no. Every segment of DNA is useful. Okay? What I mentioned to this, that making a contrast with the mitochondrial DNA and human gene is composed of exon and intron, okay? But in case of bacterial or mitochondrial, there is no exon intron system, rather ORF. This is because of the fact, space accommodation. Mitochondria is very small, they are to accommodate, so there is no intron and exon system. But they need high number of gene. What do they do? In one human cell, one pair of chromosome pair 1, chromosome pair 2, but mitochondria in one cell, that is 100 to 1000. Just they multiply the number, not size of the DNA. I would like to focus on some technical part. In cytogenetics, chromosome is studied from lymphocytes, specifically T lymphocytes. What do? We collect 2 ml of heparinized blood in heparin tube, and this heparinized blood we put in culture medium, phyto-M, and incubated in 37 degrees centigrade. Okay? We collect in heparinized blood, otherwise it will clot. And we put phytohemagglutinin 2% and FCS 15%. The question is why? You know, our peripheral blood, generally they don't divide. Normal course, they don't divide peripheral blood. But if at all our peripheral blood divide, that is the cancer cell, blood cancer. Okay, blood cancer, others will not divide. So to make them blood cell to undergo cell division, we force them undergo division, making a blood cell. We put Phyto. And for division, we need some hormone. Hormone is supported by fetal calf serum. And with this mixture, we put them in 37 degrees centigrade. We put them in 37 degrees centigrade for 72 hours and harvest following the protocol. We get this metaphase and we see under microscope. Okay? Simple, I just 1 or 2 line. In our peripheral blood they don't undergo division. If at all undergoing division, that is the blood cancer. What do? We add Phyto and FCS. Phyto means phytohemagglutinin, which stimulate the T lymphocytes to undergo division, and we put FCS, fetal calf serum, that is hormone for their development. We put it 37 degrees centigrade and we harvest this culture with following protocol. I am not going the technical defects, and we get metaphages and we screen it and arrange the chromosome. It is a karyotype. It is a karyotype. Here I have projected the first human metaphase. This man, Jew and Levan, these two scientists, first they confirm that human metaphase having 46 chromosome. But here you see I have written lucky mistake. Why? Sometimes mistake also give you some good result, but don't commit or make any mistake intentionally. These two scientists first confirmed human chromosome 46. Actually, in their laboratory, the technician made a mistake, because I did not details the technical aspect details. When we made the chromosome, we put hypotonic solution for proper spreading of the chromosome, proper disbursement of the chromosome. That technician made a mistake. He put some solution, and next day morning he met his boss and telling, "Sir, I made a mistake. What you have done?" "Well, I put this thing." And after preparation they can find, oh, very good. The number is very clear cut 46. अगर वो टेक्नीशियन गलती नहीं होता, शायद अभी तक क्रोमोसोम नंबर भी नहीं होता ठीक से. समझ गया ना? That is a lucky mistake by the technician. So mistake also give you sometimes good result. So, but don't make mistake. Jew Levan, 1956. And another story, I am coming to the next slide. You see, I have written amazing, this also happens. And these 2 lines. Can you help me? You just try to understand the previous slide and analyze the previous slide and these 2 lines. That was a mistake, and this is an amazing fact. Okay, I am telling you. Why I am telling this also happens. DNA, Watson Crick, 1953? 53, is it? अरे भैया तुमने इतना बड़ा काम कर लिया, डीएनए का कंपोजिशन बढ़ गया 53 में, और क्रोमोसोम नंबर कब किया? एक नंबर तुमको आता नहीं है, क्रोमोसोम नंबर 1956. गॉट इट? ऐसा भी होता है. हाँ, फर्स्ट थिंग ही मेड द मिस्टेक, गॉट द एक्चुअल नंबर, and ultimately DNA you put it 53 and chromosome number is 56. Okay, one thing you know, chromosome biology is much more difficult than the molecular genetics of DNA study. Why? DNA, you just from the chromosome you kick out the protein, you are getting the DNA, set protocol is there, NGS is there, sequence will be there. Okay, but in case of chromosome it is not that easy, because chromosome means, as I have shown you nucleosome model, chromosome means histone octamer wrapped by the DNA, and I have skipped so many things, linker DNA, okay, and nucleosome model, solenoid structure. What for? To accommodate that 2-meter DNA in a 5 to 10 micron of nucleus. Nature has made this protocol. To handle that, when you are handling this chromosome, you are handling that protein, DNA, everything. So, as if molecular genetics is very difficult, no, it is easy. Kick out the protein and automation DNA, but protein you cannot. Why? I will tell you again. Whenever we do the chromosome identification or karyotyping for disease diagnosis, same day 2 culture, one patient is giving the result, another patient is not giving, because we are not getting the, in one patient we are getting the proper G band, and another it is not responding. Why? Actually G band is a just profiling of euchromatin and heterochromatin. Euchromatin and heterochromatin. So on that basis, we facing the processing of G banding, because we are dealing with the chromosome. I will come. It is a karyotype of male XY. Rather, I have to go fast, I believe so. Okay, I have shown number of slides, but I will go very fast because time constraint is there. It is female XX and it is G banded. G band, that is whenever we try to identify, we use the G band. And different types of chromosome, metacentric, submetacentric, acrocentric, but not telocentric. It is in mice. And I use the term chromosome dynamics, chromosome dynamics. This is chromosome dynamics, again you see. This structure you can see only during cell division. That is kinetochore. This kinetochore appears during cell division because it has to be attached with spindle fiber and pulled to the polar region. And this is the dynamics of chromosome. And how this is happening and what sensor is working, very difficult. I was telling you bar body in the nuclear region and at the periphery of nucleus, this is. And different types of bending process we use for identification. Okay. Different types of bending process we use for chromosome identification. Here you can see G bending process, here Q bending process, here C bending process, and NOR, nuclear organization. Nuclear organization is a very vital part for rDNA, rRNA, ribosome synthesis, and protein synthesis. This is the NOR region, and this is telomeric DNA, and telomere playing a very important role in our cell division process. We know that during cell division some 5 base pair is lost every time, and that determine the how many times cell division has undergone. If you remember, I told you the chromosomes are not randomly placed. Is a particular place. If you take human chromosome, it is 18 chromosome near to the then within 15 minutes I try to next one. Hello, just give me 15 minutes time. Give me 15 minutes time. You just what I told about the chromosome, I'm making a one sentence, you read it. That is, in the nucleus, chromosomes are not randomly placed. There is a natural order which chromosome which takes at what point and how they will be organized. Got it? Just you go through these 3 sentences, you can get it. That is, chromosomes are properly placed in a particular place depending on their functions. Okay? Here. नहीं time I have 10 minutes time. Hello, I was, if you remember at the initial I was telling you putting the cell division, some problem in the cell division will give rise to syndrome. Just I am going to, this is the Down syndrome, you can see here and there, the trisomy 21, this is a problem with the mitotic process. This is in hospital, medical colleges, we very often get this type of syndrome patient, that's Down syndrome, trisomy 21. You must be knowing about this. You see, I am very interested for this. In our medical college, in a single day, 3 patients was referred from pediatrics department, and we got all are Down syndrome. That is trisomy 21. And trisomy 21 may be segregated, or 3 are separated, may be translocated with 14 number chromosome. Not, हाँ जी, हाँ बोलिए, हाँ रॉबर्टसनियन ट्रांसलोकेशन नहीं है, correct, and it is Edward Pato syndrome. It is Pato syndrome, chromosome number 13. And here you can see very classical Klinefelter syndrome, 47XXY. But here you can see the same clinical feature, but it is not a Klinefelter syndrome. It is hormonal problem. Sometimes they are reporting to the surgery department with a feature of Klinefelter syndrome. But when you do the karyotype, we see it is not a problem with Klinefelter, rather hormonal problem. Sometimes we can find XYY. In case of female, when they attain menarche at the age of 14, 15, they are not their period, they are referred from gynae department. We can find different types of abnormality in case of female. Turner syndrome. It is mosaic Turner. Mosaic Turner. This type of female also you can find if primary amenorrhea, not attain menarche, or recurrent abortion, we can find they have some 3X chromosome. Is a Turner 46XX and XXX. Here you can see again from different hospital ward, they find male. We can find 2 types of syndrome mainly. One is Klinefelter XXY, another is XYY. And interesting thing is, this Klinefelter is no fertility, but in case of XYY, sometimes they are fertile. In case of female, Turner is infertile, but XXX syndrome, they are fertile. But they have some sexual problem. And these are some translocation in case of primary amenorrhea. This is patient referred from again primary amenorrhea, this type of XQ translocation. Here you can see a very interesting thing, that male 46X0XY, male. Why is there? Male is okay. But if you see the same karyotype from OPD, they referred the same karyotype, it is a female. What happened actually? As I told you, during gametogenesis or oogenesis, there is a problem with the crossing over. XY is translocated to Y chromosome. XY, Y, SRY translocated to X chromosome. So you can get this type of problem. Here I will show a number of slides, which are those they reported in the gynae, recurrent abortion, and you can see a reciprocal translocation. It is not necessarily the translocation from the female, sometimes male candidate XY showing the translocation. As we know always, XX is male, correct? XX is female, XY is male? Not necessarily. Not necessarily. When they are attending the OPD, some problem is there, they are referred to cytogenetic laboratory, and we can find, you see, here is male XX. How? Male XX, because this X chromosome acquired the SRY during gametogenesis from the Y, they acquired the SRY. Here is female, not XX, XY. What is there? The Y lost the SRY gene. Y lost the SRY gene. So without SRY, it is a female. This is male pseudo hermaphrodite. Karyotype is XY. External genitalia is female. They go to the gynae department as a female, not attendant monarchy. When we do the karyotype, we can find that is a XY karyotype. It is a hormonal problem. Moreover, one thing, just X chromosome or sex chromosome, not for sexual characters. These are for somatic characters as well as sexual characters. What I am telling, sex chromosome, X and Y, that is responsible for sex characters. No. In X and Y chromosome, there are genes which are responsible for sexual as well as somatic characters. Here is a test-specific gene on the Y chromosome. Here, X homologous gene. What I mean to say, in case of female, 2X chromosome, one is inactivated, okay? But not all the genes are inactivated. Some genes are still active. Which are those? Those who got the homologous genes on the Y chromosome. Those genes on the X chromosome got the homologous genes on the Y, they are not inactivated. Some genes are still activated in the inactivated X chromosome or bar body. This is what I already told. You see, it is a male XX because acquired the SRY. This is a female XY because no SRY. Just take 3 or 4 lines. Nowadays, in medical colleges, human genetics laboratory is must. That is advisory note from the MCI. They are looking for young human or medical geneticists. They are not getting trained personnel. Okay? So, because of the fact now in India they understand all the diseases and disorder, the main fact lying with chromosome, gene and genome. So, to understand diagnosis, treatment, prognosis of the disease through gene genome is must, which is coming nowadays as a science, personalized medicine or genomic medicine in medical colleges. Simple, our body system depends on chromosome, gene, genome, abnormality due to gene, genome, disease diagnosis, Nowadays, doctors depends on gene genome for chromosome diagnosis and treatment prognosis, personalized medicine, genomic medicine or precision medicine is coming. Of course, in India, we are lagging behind. Thank you.
Thank you. I think Sir has delivered very fundamental and advanced level of genetics and cytogenetics part. And now I'd like to request Professor Dr. Bulganin Mitra to deliver a vote of thanks. Sir.
Thank you. Thank you, Dr. Ajay, giving this opportunity to say something about Dr. Pal and also the last portion of the vote of thanks. So I do not know how many people, how many students you are knowing that what is the relationship with Dr. Pal and me. Actually, we are very close friends. We met in our science college in our postgraduate days. So that time, from that time, we are friends, but different field. In genetics, I do not know, I do not understand also genetics. That is the difficulties with me. But something is similar with Dr. Pal. That is, both we are loving this Ramakrishna mission. Only for this reason, Dr. Pal came from Maharashtra to Kolkata to say something in front of you. So I must thank Dr. Pal. It is your great, generous. I appreciate everything. So last, I from my side and on behalf of Ramakrishna Mission, I am thanking to you that you personally came here with your busy schedule, and you do not know that Dr. Pal actually used to reach Kolkata on fast, but due to some health problem and some family problems, he came late here. But he was definite that he will come to Ramakrishna Mission, our college, and will deliver this talk. So again, I am thankful to Dr. Pal. He came here and giving this his generous, that's the burning topics of the human genetics in front of our students. And I think he has told the first line that I am here to motivate you. I think our students are motivated, sir, from your lecture. It's a very useful lecture for our students. So thanks and thanks and thanks once again. I am thankful personally Dr. Ajay Mullik, because he is the man who is behind of this seminar. He is a totally he is engaged himself to communicate with Dr. Pal, and ultimately he has succeeded for these things, and I hope near future Dr. Pal will come and Dr. Ajay Mullik will do this everything. So thanks Dr. Ajay Mullik for your this seminar. I thanks all the students who have participated this meeting, and I think some knowledge you have gained. This is a take-home message, like these things you are taking, and in future it will be, if you want to meet or you can contact with Dr. Pal, it is free to access. So I, on behalf of Ramakrishna Mission, I thanks everybody who are directly or indirectly involved and help to success this seminar today. Thank you everybody. Thank you.